we'll start here. And the first module, marker data profiling, is in MicrobiomeAnalyst. So basically, we will use this module to understand the community, the microbiome community. What we are going to know after analysis in this module is basically we need like the metadata you are interested. For example, in this example data, we are looking at the difference between the IBD, like the subtype CD group, with the control group. So we want to know what's the different microbe species between these two groups, and if there's a overall different pattern between these two groups, and the dominant species, and if there's correlations between these species, that's what we will get to know after analysis. So we'll start from here, and here is the input table. I will introduce this on the website to you. So you can download the files used in this module in our GitHub by this link, or if you have interest in the raw data processing, you can directly process and get the files directly from the result of that module. So let's work on the website. So you just click Start, and then click this module. So you can see that MicrobiomeAnalyst actually accept different formats of the input data to avoid make you too overwhelming of the data format. Here I just simply introduce the basic information we need to upload in the website using the plain text format. So here you can see these ones, except the phylogenetic tree, all these three files are mandatory. First of all is the ASV abundance table. So from the raw data processing, in last section I have shown that we got the sequence, you can see the ASV sequence, not only the ID. So with that sequence abundance, we can check here, and later this is for the functional prediction part. And so here we just choose the file we get from the raw data processing. Here you can see this is a sample ID, and then there is a sequence, and then we have the abundance. So we just need to take attention that the first column of the first column, the name of the first column must be the capitalized name. This is for the recognition of our website. So you just submit the first file, and you can just hover here to see the example data. So always format your data like this. And then second important information is the metadata. So you can see here the metadata. The first is also the name column. It's a sample name which should exactly match with your ASV abundance table. And then you can have several columns, like the group, the age, and the whatever meta information you would like to include to treat as covariant later. So here we just submit what we get from the raw data processing. It looks like this, same as the example. And then the last is the taxonomy annotation. The most different thing from microbiome data with other omics is it have 7 layers, different taxonomy level. So we need to upload the taxonomy annotation. So here, the first column should be the ASV sequence. This sequence should be exactly same order and same as your ASV abundance table. You need to format it like this. But if you use, for example, the DAT tool processing our website, this will all automatically format. So you just submit. So these 3 files are the basic information you need to input. No matter what kind of format you use, maybe biome or mother, if you know about that, the core information is always like the abundance and the metadata and then the taxonomy. So here we don't have a phylogenetic tree, so we just escape this step, and then we need to select the taxonomy label. So just now we use a Silva database, so we just select it, then submit. Now we get to the sanity check page. Here we get an overall of our microbiome data. So we can see exactly what we have input, like the how many samples and how many sequence. And then if there's a singleton data and the summary of your metadata and the minimum, maximum counts for the sample. And then if all the samples are matched. So here is like everything is all right for the example data. If you see some, like if your data is not totally matched, so we will have a error message, then you need to reformat your data. So here we can see the first row, there's a default filtering. So we usually remove those features that only appear in one sample because we think that's maybe a noise or it's not that the main feature we are interested in. So you can define the singleton you would like to study. For example, if you think in total it only have one count, so you remove that, it's also okay, you can choose that. Or you want to, sometimes your sample is focused on the rare species, then you have no need to remove the singleton. So in this case, we just keep the default. And then when you change the tab to the meta overview, and then you can see the metadata in your data set. So we have basically 3 columns. The first is the diagnosis. It has 2 groups, the CD group and the control group, then the sex and the age. So next we can, here everything is all right. We can then proceed to the next step. Then we go to the data filtering, because in this statistical analysis, we basically need to do the comparison. So we have the primary metadata. We require you to have a category metadata like this diagnosis or treatment. So we want to remove those data that might be low count. If it's the count is quite low, it might be the error, sequencing error, or it's not the main contributor. And then if the data have very low variance or it's constant to all the samples, then it's not meaningful for our comparison study, so we can remove those. The detailed explanation is here, and we keep the default mode and submit to remove. And here we can see that there's some low abundance and low variance features has been removed. And then we process to next, and we got the data normalization. And we have 3 types of normalization. First, the data referring. This is for subsampling to make trying to make the sequencing more comparable. And then data scaling, this is more to address the library size between the samples. And then the data transformation, it's this method is more have some backend assumption, and they are trying to address the compositional of the microbiome data, because the specific of microbiome data is it has a lot of zero, and it's compositional, as Dr. Xia mentioned before. So each normalization method have its own assumption, and we use this default one is the most like a common one. It address the to address the library size and remove the effect of the dominant species, and this is more suitable for exploration analysis. So here we just keep it the default and submit. So we proceed, and because we didn't do the rarefaction curve before, we can check if our data need a rarefaction. So you can see here, most of the samples are reach the plateau stage. So which means it's more or less okay, except there's this one. It seems not sequencing well, and you can go back to the normalization page to do a rarefy, and but this is not mandatory, because sometimes if you do the rarefy, it will we have lose some information because we subsampling. In this case, we because we have only this sample, like it's not quite satisfied, so we keep it like this. And then we have this analyst overview for different functions, and I will pick some like we have discussed to show the main features of what I can get from the microbiome data. So first is overall, it's the visual exploration of the patterns. So you can see here we got the stack bar plot of each sample, and we can compare between the different groups, and you can see we can change the taxonomy level, maybe species, and yeah, we can make it to the relative. You can change here to see the relative abundance. So you can see a lot of are not assigned. It's common because it's 16S data. So you can see each sample, their structure, if the overall, if there's different. And we can see like most species appear in all the groups, but maybe they have different percentage. And this is a visual exploration, so we don't have a statistical control. Then we can use the diversity to see their statistical result. Like the first one is the common alpha diversity. We provide like a different method to show the observation or the evenness, richness of the species. So here we use first is this should be close to the observed. It's consider the category, and we can see there seems a difference between the CD group and the control group. Control group seems to have more diversity. Then we have the statistical method, like here we use the Welsh t-test for the post-hoc comparison, and then we got this. It seems the p-value it's close to significant, so there might be a difference in these two groups, and we can also look maybe the Simpson to consider more complex thing. If we do this, it's like we see it's more close, and you see the p-value change to quite large. So it means maybe there's some dominant species, and there's not a big difference of the evenness, but there's maybe a difference of the richness of between the groups. And then we go to the beta diversity. This is the overall pattern to see if these two groups are different. As we can see here, they have a lot of overlap. We have we provide different methods like PICO-A, PCA, and NMDS method and distance. So you can using the question mark to look into the detail with the different, and you can try different methods for exploration. And we have this statistical method to tell us if the group variation is significant. We can see here, it seems that the two groups didn't has too much difference based on this method. This is actually consistent with the literature. So even though the patterns, the overall patterns didn't quite separate, we can still look into more individual features that are different from the two groups. So we can also color the dots by different, like for example, the alpha diversity. So you can see the pattern is more clear here, and the alpha diversity distribution according to the patterns. So we also have the interactive 3D plot to look into more dimension, and if you have more groups, more than two groups, then here we also provide the pairwise statistical to show the significance of the result. And you can also change the experimental factor if you have more than one groups to see if they have difference. Oh, I should do this. But in this example, we are more care about the diagnosis group. So next, we will do the comparison analysis. As you can see here, we have different methods, the single factor, the multi-factor, and the left side, and the random forest. Random forest should be more common in different omics. And we have the single factor statistical analysis for comparison of the different groups. So we just compare. You can select a different taxonomy level, and here we have like 6 method. We have the basic t-test and ANOVA, and then we also, but the more widely used, we use HR because it's there's a review showing that HR catch a better difference between the groups, but it may have some false positives. And all these models, they have some, they are dedicated to the microbiome data to address the zero count or the compositional structure. And so we just have an example. It's hard to tell which one is the best. So usually you may try different methods and to see those stable features that consistently different, then that might be robust. That's indeed different from from the different groups. Here, the different color showing the higher taxonomy level. Here we use the phylum, so each color showing us a specific phylum, and then here the dots show the species that significant differential. So you can click and to see the box plot. And if the triangles are like upset, which means it's like it's increased in the reference group, and we can see a lot the triangles down face down, so they are decrease in the reference group. And we can see the result table here, and the highlight rows are the features that find significant, and you can click the logos to see the box plot, and also you can download all the figures and tables here. This is for the single factor analysis, and more commonly used method is massly, which it can consider the covariant. So here we use example. For example, we use sex and age as covariant, and then we submit. So we can see here's no significant features because we use a linear model. Linear model assume that the data was normal distribution, but it's usually not the case for the microbiome data. So we may find other models. This more specific to the microbiome data. We can see if there's significant features. So you see, there's 5 species that show up, and the green ones are significant when we adjust it, and the red ones are the features that non significant when we adjust. And so because most 2 out of 3 features become non-significant when you adjust it, so you need to consider if this model is correct, because the significant is relative small, so it's a little bit hard to tell the model. In this case, we may think about if from the biological side, if the covariant we are choosing is meaningful. So this is a full differential analysis, and we got the result table. It's similar as a single factor. And
can I ask you a question?
Yeah.
Just to be 100% sure I understand it, adjusted, you say adjusted versus non-adjusted.
Yeah.
Are we talking about multiple comparison adjustment or the adjustment for the covariant?
Adjustment for the covariant.
Okay, so
yeah.
Because it says control for, but it's actually, we can
yeah.
also say adjustment for. So it is the blue versus the green, for instance, right?
Yeah.
The blue is without these covariates.
Yeah.
And the green when you put them into the model.
Yeah. I can show you if we don't have covariant, we only have the blue ones. You see, okay. You have only 3 features here. Oh, here it's overlap. Then when you have covariant, you can show you see, there's two features are not significant anymore because I only include one, so it may be. So this is the adjustment, which means, and here's the FDR if we got a p-value.
Is this the one method for the multiple comparison adjustment, which is FDR?
Yeah.
Okay.
Yeah. So.
Thank you.
Yeah.
Like whatever result you are getting, are these similar with the original paper? Have you compared your results and the results originally given in the paper are equivalent or same similar kind of?
I didn't compare because I just use a subset of the data and
because like reproducibility is a key. So if the data is reproducible as similar to the original paper, so like maybe more beneficial.
Yeah. I didn't check it because the microbiome data and the metabolomics data in the database is generated in different years. So I'm not quite sure if they indeed have a paper like the original database. They have a paper. They have a multi-omics paper, like you can see in the slides when we introduce this data. But I'm not quite sure if they indeed use what I use this data because not all the data has been published. So, but according to the pattern we got, like for example, this species is actually reported, even it's not using this data, but it's consistent with the previous study of IBD. Yeah, like the pattern, like the microbiome, it might not quite separate between the groups, but there are always some features like underlie signal that they make sense. So I'm running out of time, so just the last one. So for the function prediction, so before we are just looking for the computational information, so we have the functional prediction part reaction, the function for predicting the functional capacities. So here's the result because it takes some time. So you can get the KOs from the sequencing data. So this should be the all the information for this section.
So thank you for the questions, and clearly reproducibility is what we focus on. So when we initially publish, we compare with Hudenhauer group from the Human Microbiome Project. So for example, metabolomics, we're doing better because we are specialized in metabolomics, and we see a strong batch effect, but overall the biological conclusion stays because the function is actually very robust. And in terms of microbiome, a lot of the things we have done, like Maslin 2, and is follow what they have done. So reproducibility is actually a baseline for what we are doing. We actually, because we follow what they have done and also adapt to the latest result. So most time, our result is better. I will see that. But again, it's always keep biology in mind, and we realize that a lot of times specific normalization filtering approach at the very beginning have some effect in the later. So sometimes you click buttons, accept the default. You didn't realize that have a downstream. You said, hey, Jeff, I couldn't get the same result. Then we said, can you share the script? And I realized at the beginning they choose a different option. So all of this is really speak to the concept. Some of this initial data treatment do have a effect most time, but the robust overall pattern of function is very, very robust, and we can see that. It's just in a demo before when Yao did it, and you can see alpha diversity. We do not see a lot of difference. The p-value is just marginal. When you choose and become very not so significant. And the beta diversity, the same thing. And we do this comparative analysis. And all of them actually speak the same thing. They are not hugely different, but there is some patterns. So it is common in clinical. You do not see a huge difference. But more or less, they're telling you the same thing. If one approach is give you huge different patterns, and all of the other one actually speak the other story, and you need to be very cautious whether it's artifacts or actually a true discovery. So overall is that, they are consistent. We have different approaches talking the same thing. Visually, you see there are some patterns, but statistically, it seems marginal, not so significant. Then you try different things, but all of them give you the same thing. At the moment, which one working best is you as an expert in the domain, you try to leverage your knowledge, leverage the literature, and try to come up with the whole, how you describe the whole story. So this is something become more important when we go to multi-omics. So sometimes multi-omics is really strong, tell you a story. Sometimes it's very subtle, and some people can really, from these patterns, come up with a good explanation, but a lot of times it's tricky, especially for us. If we are not a domain expert, we are very cautious about seeing anything, but we try to give you a different perspective of the same data. So if we talk about multi-omics, but now focus on microbiome and metabolomics, and there's different combinations. You have marker gene table, you have shotgun, you have a gene abundance table. So this is just one marker gene, 16S, 18S, this is all the genes. And if we do a target metabolomics, it's metabolized conjugate table. On target is LCMS peak intensity table. So we have 4 different type of tables, and you can pair any of them, one yellow and one green. So you can do the pairing. How do you integrate them, right? So there's something that's more universal, disregarding what type of data. You statistical-wise, you can always correlate them. So correlation analysis, a correlation network. You can do a univariate, a feature correlation network. This is very common, and the only thing you need to be cautious because it's high dimensional. If you're doing this, you could get a lot of false positives. So you need to looking for major patterns rather than just individual significance. You need to really zoom out and feel it, see this one, not just focus on particular significant cutoff, just because univariate correlation analysis on the cross-omics is very is useful, but it's tricky. So you get a lot of stuff. Then the multivariate cluster analysis. If you are familiar with the machine learning, precision medicine, and the multi-omics, then there's a similarity network fusion, spectrum clustering. So there are several different things. You identify some cluster, similar samples close together. But why they're similar is usually non-linear. So this is kind of SNF, or spectrum clustering is not linear. So it's very hard to, intuitively understanding why, because the underlying feature is not a linear one, but it's very robust, the same good performance. And we do have tools called Omics Analyst. We provide a lot of things, but it's not, today we are not going to focus on that. We actually focus on the univariate and multivariate, which we call the procrusting analysis, which is relatively widely used. Then there's Diablo also doing analysis. They are more linearly combination, try to explain the covariance. And so for the machine learning, we are not going to cover. It's just because, not because they are not useful, it's just that we do not have time to cover them. Now, we also can do functional integration. Functional integration is using metabolic networks, and we can project data into this metabolic network and see the genes, and using the 2 different omics and project on the same visualization and see are they telling the same stories, So functional-wise, we have CAG network, metabolic network. We also have genome scale metabolic network. They are, you feel similar, but they are slightly also different. And so they are, especially GEMs, they are more computationally friendly. CAG is more or less a visualization and according to our own knowledge because it's manually curated. But GEM is a lot of the system biology built in. So now here we're talking about statistical correlation. You can do univariate. This is a univariate correlation network, and you can do multivariate. This is canonical correlation analysis. So it's multivariate foundry covariance, so CCA. And more or less, you think about PCA. PCA is one matrix and explain most variance. If it's two matrix, you're doing CCA is explain most covariance across two big matrix. So this is PCA is one matrix, and CCA is two matrix, how they covariate with each other, okay? This is just intuitive like this. How internally doing it, because linear, actually you can understand, but how useful it is, it's really case by case. But here I show you the multi-omics correlated network. We can do the correlated network among metabolites, among genes, or among metabolites and genes, among metabolites and microbes. So this is very easy. And the only thing I just mentioned is about how you control the threshold. Otherwise, everything could be correlated. A lot of other things purely by coincidence is false positive. And it's not in MicrobiomeAnalyst. It's called omics analyst. We allow you to control at a different level. So you can see within the omics and the cross omics, some of them is very interesting. They form a very different topology, and they are so tightly clustering them. And one of the other omics create a bottleneck and actually leverage the things. And it seems very interesting, the shape. And so for us, it's from data science point of view, and it seems very interesting. But biologically, it's really based on your particular field. So I'm just leaving it to we can do enrichment analysis on this network. So what I would like to say is you can easily generate an overwhelming correlation network, but a lot of them could be just random. So really you need to control certain threshold and form something more robust. So a multivariate covariance analysis, I just mentioned about individually you can see their trend by PCA or PCA. Individually you already feel it. But two omics together, how they correlated. Visually you try to think, but CCA, there's Diablo or MOFA, this is statistical method, explicitly trying to teasing them off. And we have omics.analyst.ca to explicitly doing that. But for microbiome analyst, we try to do is Diablo and pro-crust analysis. So this is a really visualization plus that is why we choose this is because most widely used and relatively easy to understand. You want more and you use omics analyst. So we are so pro-crust analysis. So they force two omics try to align with each other. So and here is that I just found out Wikipedia, they force two things, for example, bad and the person. If your leg is too long, you just really stretch or cut it off, make it try to fit. And this is really talking about metabolomics and the microbiome. Or with transcriptomics, you have 25,000 genes, but you only have 1,000 of the metabolites. How you to align them? And somehow one of the omics need to be stretched, aligned, rotated. Finally, this seems best aligned. At the moment, you try to see what's the correlation or what it looks like. So this is something statistical, semi-intuitive, but how you're making sense is different. Really, you need to use your own domain knowledge. And why we choose this is because more or less Rob Knight, who was the pioneer in microbiome studies and also in microbiome metabolomics integration. And what they try to do is try to really combine them to have a faster maybe diagnosis or precision medicine, and so try to have multi-omics. So this is what they try to use just the pro-crust analysis, and they're able to find that they are very specific, environment specific, different household, different background, even they have very unique configuration, and which is actually not that surprise because you have different lifestyle, and that one affect your diet, affect your microbiome, and environment survey, everything combined, how you integrate them, and they actually use CCA and see that. And Diablo is another approach which is explicitly consider two matrix and also consider this major factor, for example, control on disease. So it's supervised, but also consider explain the covariance. So it is basically, you can see the CCA is taking about 2 matrices, try to make them correlated. But Diablo plus that Y, a single Y column, control and disease. So they make it more like explain variance, explain covariance, and explain the difference, basically. So that seems making more sense. It seems more powerful. So this is a view, and if we use omics, I think this one is in the MicrobiomeAnalyst. So you're probably going to the demo. So you can just control how much covariance you want to explain, or how much difference between two groups you want to complain. You want to explain. So they allow you to actually identify these core features that multi-omics features that explain covariance and also explain the difference between different groups. So we're talking about, before we talk about the statistical integration, univariate, multivariate, okay? Now we're talking about the functional integration. And there's 2 approaches. It's a metabolic network and gem-based. So we just mentioned, now we're talking about the metabolic network. How do we do that? Is that first we want to use microbiome data and use metabolomic data, all of them in the same brain to the same page, okay? Where is the same page? This page is called the KEG metabolic network or metabolic network. How do I do that? If it's a marker gene and you have marker genes and you actually can identify these microbes, and these microbes actually have their metabolic reaction networks. We can use them when they're present. We keep their pathways. When they didn't detect it, we just remove them. So this marker gene actually help you to contextualize the KEG network. Then you can project your metabolic data, and together you jointly speaking to the same thing. And this is try to refine your background, and then you overlay with your metabolic network, metabolites, what you measured. And if a shotgun metagenomics, you actually measure the genes or enzymes, and you have, the other side, you have metabolites. This is most straightforward. You know, you MetaboAnalyst, we actually allow you to have proteomics or genes together with metabolites to join the pathway analysis. So the same thing, we can do that. But here is actually, we can actually using the other part, we can actually use a genome scale metabolic model to predict how likely they can generate metabolites. So here's something we call the contextualized metabolic network. If we know certain species is present or not present, we can actually filter the general networks. Something pathway reaction will be present because you detect them. If you didn't detect them, make it a gray out or totally hide them. Now all your current metabolic metabolites is overlay on this customized network, and you can actually see the compounds and reactions and highlight these significant correlations. And you actually click on it and see which compound, which microbes have capacity to generate compounds. And this capacity we can actually predict using a gem model. So overall, we leverage the knowledge about their known metabolic capacity, and together with what we detected with metabolites to really help you see whether there's a link and what's the hypothesis. If you're able to culture these particular microbes and validate it, then that's your high-profile paper. And for us, this is what a computational work can help you. Just use multi-omics data, refine it, and pinpoint a few of these microbes, and some of them hopefully you can culture, or somehow you can follow. Then you can identify whether that microbes produce that metabolites. So if we use genome scale metabolic model, we do have an understanding globally how what's the metabolic capacity, and we can actually predict how likely they are generating certain metabolites. Okay, before we already talked about pie crust. You have these genes and the stuff, but we can actually leverage that from the genome to metabolites. Okay, you have the genome capacity, you're going to have capacity to generate metabolites. But we are not using CAG, we use GEM, which seems more accurate. And again, as we found out, the species level prediction is most accurate. And if you go to too low or too high, it became not so good. And you can want to get 18%, but if you get the 16%, it's not that useful. So genus level using marker gene is very useful. So we can actually combine both of them and statistical correlation overlay on what we can predict. You think they are correlated statistically, these metabolites microbe co-appear, but are these microbes actually most likely to generate this metabolites based on the GEM genome scale metabolic model? So when two evidence, one is from your data correlation, one is from your knowledge-based prediction overlap, the confidence is higher, right? So this is something we just allow you to merge different evidence, and this is just a simple correlation network, and on top of them, and you overlay what is the probability. It's that metabolites can produce that. That microbes can produce that metabolites. So you overlay two independent evidence, statistical correlation and GEM-based prediction. So some of them will overlap, and some of them are actually significant correlation plus GEM-based prediction. Both of them point to the same thing, and that microbes, that metabolites deserve your attention. If you want to culture them, you want to purchase the compounds to do validation, and that's most likely you'll be successful. Otherwise, it's going to be too wide, which one you're going to focus on. So this part to help you to pinpoint and just reduce your target list. So the next one is actually we talk about the metabolomics and the microbiome and how they correlated using the data. And here is that we actually want to see, can we actually find their mechanistic interactions? And so we know we can get a GEM and get their metabolized potential. And we also have large scales. We know the association networks because a human microbiome project, and together is a lot of the studies. They have the microbiome. They are highly associated with each other. But why they highly associated with each other and how they interact? And sometimes we know, because there's a food and they all like to attracted to the same food source. They are not interacting, which is fine. We know it's probably just purely being together because they have the common food source. But some of them is more actually interact, and which is have a small proportion, but is highly interesting because they actually compete or complement each other. And this one actually we can formally test and actually predict in a large scale. And from this large number of association or correlation network and plus GEM, we can see, they are co-appearance, most likely because they compete or complement the same type of compounds. So in this case, we use metabolites as a mediator, communication between the two microbes. So which is, we believe, theoretically, is how they usually interact. And this is, if we want to do this, we need to understand, we need to really leverage this GEMs, and we need to identify what these microbes need in order to survive. And this one is called the seed metabolites. So they need to obtain this nutrition from the environment to survive. So if this is, we can define this requirement, nutritional requirement, and then we can compare this profile among different microbes. If two of them have the same nutritional profile, and they are most likely compete with each other, right? If one of them actually produces metabolites, which is required by the other one, they are complement each other, right? So this is intuitively making sense, but practically, can we do it? And so this is something really triggering, triggering our development work. So So this is how we would like to develop microbiome net. So we have millions of the correlation or association networks detected by the literature. And from there, we would like to see how their metabolism, how their compatibility, how they, and actually we can identify some of them. It looks very interesting, compete or complement. Some of them is random. You see they do not like each other, do not actually interact. They are more or less normal or just strangers, they do not. But some of them is really, really interesting. And when you see this very interesting things, you can, you probably, there's some metabolic interaction between them. And you can zoom in from this GEM networks and see the pathways. So this is the whole workflow. And this is a microbiome net we try to help. And clearly, this is, it's for 5.8 million microbial statistical association from 76 studies. So this is a huge one. From there, we would like to see how their correlation, but what type of correlation actually can have some mechanistic interpretation. And this correlation is identified from different environment, human gut, soil, and plant, so a lot of things. And this is come up with a neighborhood map. For every microbes, you can actually find out all those microbes we have the GEM. We can compute their distance. And this is metabolic distance. So how similar or how complement their metabolically metabolic capacity. The other one's taxonomy distance. So this one's the far away probably more different or more complement. So more close is more similar. So if you remember at the beginning of my slides, we talked about taxonomy and function. They are very different. So but they are real. So we need to consider both. So here is actually we use the taxonomy. We use the metabolic. Actually, just using a scatter plot to see how they looks like. And for example, here is one of the most famous example. It's I cannot pronounce. It's prosnicid. This is fragilis. If we do this, it's complementary index, and this is 1 minus competition. So the competition, because we have metabolics, we can see how they cross-feeding is complete, or if they compete, and we just 1 minus compete. So it's basically we still want to see the further away they are more. So we want to see that around this direction they are more likely to be have a same story. So for example, here is that very interesting. We see the standing out. Majority they probably a lot of a community have a similar distance. But some of them indeed very unique. And here is a GEM model, and for me it's very complex. So we actually can identify the regions that really a lot of them looks different. Actually here, because of two species, we can use two different colors. We can zoom in. This is really enriched for the complementary analysis. And this is actually, you get the pathways out. It is starch degradation. And you can see there's a lot of complement pathways. And this is actually well documented. So not surprising, but for us, it confirms whatever you compute is actually can help identify those complement capacities in their metabolism. So I guess we are ready for next demo. But at the moment, anyone feel free to ask questions. Yes.
So I have a question. I might have missed it. So the integration which we are talking about, is it the integration of the data acquired on different samples from the same person, like microbiome data, for example, from saliva, and metabolomics data from biofood, like serum or plasma? Can it be integrated, or will it make sense to integrate them?
The tool is, so when you do the data analysis, you upload the data, but how you interpret data is decided by what do you think is biologically feasible? I mean, saliva and blood you're talking about? Plasma, right? Serum.
Any other biofluid or any other.
Yeah, so if you think, so first sometimes we want to just go crazy and see whether there is some correlation and if the correlation making biological sense, right? So if you are the expert in the field, you think disease diagnosis actually can be reflecting on the saliva rather than using the blood, then there must be a connection. But what's that connection? Is meaningful or not? You need to think there's a ways for the small compounds actually can go through systematically. They can cross blood-brain barriers or stuff. Then if there's such a plausible mechanism, you're fine. But sometimes even you don't know, but you find it correlated very well, then it's a new opportunity. So I myself is, why you collecting data like that? There must be a reason. You suspect it might be possible, right? So the tool itself is definitely not going to prevent you from doing that, but it's your decision how you're going to defend yourself if you find they are correlated, and how you give a hypothesis, right? But microbes and metabolites is actually can travel. We have saliva, we swallow to the digestive system, and sometimes you can go into blood, and sometimes take multiple steps, sometimes it's probably more close. And so, if you have strong patterns and correlations, and you give the hypothesis, and establish a mechanism, and you can name something as your name, so it's a very good discovery, right? But data is really neutral, and you have two matrix, you just do analysis, and that context is very important to write the whole paper. And so I myself am not an expert in the background which one you have in your mind, but overall so you have the total control, right? Yeah, so good.
So in this section, I will do the demo for the data integration. We will have two parts. First part, we'll use the MicrobiomeAnalyst, the MicrobiomeAnalyst module, to do the integration from the two data matrix. Then next, we go to the MicrobiomeAnalyst. So first, we start from here. And you can also see the GitHub, the link that get the tables we need here, or you can do both raw data processing, either the metabolomics and both the metabolomics and the microbiome data, and then you will generate the tables upload here. Here in MicrobiomeAnalyst, when we do the integration, we need a metadata file. So the metadata file should be exactly same for we only need one, so the samples should be same in microbiome data and the metabolomics data. So make sure when you do the raw data processing, and the raw files you generated from the machine, maybe the name is different, but when you upload here, you need to make the sample names consistent from the different table. So we can see here we need three types of data. First, the metadata. This is the shared one for both omics data. Then for microbiome data, we accept either the OTO or ASV count data, or we also accept the result from shotgun metagenome data, which can be a KO abundance table, but we don't demo it here. If you are interested, you can try our example. You can see we list a different type of data here. You can try anyone you are interested to see the difference. So for the metabolomics data, we support both targeted metabolomics data and untargeted. So if you have annotated data, then you can you get the compound name, and then you can use the targeted one. If in our example, we actually don't have MS2 data, we didn't annotate the peaks, so we only have the peaks, then we can choose to use the untargeted data. So here I will start from uploading the data. Then we choose the metadata is the same one as the marker gene module. And then the ASV count table, it's also the same as the last section. We have the ASV sequence abundance and the taxonomy annotation here, and we specify the taxonomy labels. And then for the untargeted metabolomics data, when you see this, if you use the MetaboAnalyst, you should find this is the same actually. So first, we should note that our data was negative, generated from negative mode, and we use the default ppm tolerance. And then we have the retention time, and we can upload the peak intensity table, and you can see the intensity it looks like this. It's just change the feature names to the peak names. It's the other part is all same as the ASV table. So we just choose and submit. This may, when we uploading, may seem there's a lot of stuff here, but you can only focus on the data tab you are interested. And process, we will also go to the summary page to see the detail in our data set. We got the microbiome data, metabolomics data's feature number, and also the sample number, and the primary metadata groups. And also, we also have this same as marker gene. I won't repeat it. And also, you can see the metadata overview. It's also keep the same. You can always edit the metadata. I forgot to mention if you want to change the factor, the order, or you can change the group label if you find any error. So, or you can remove any metadata. It's not necessary. You can always add it here. So here we get everything all right. So we can just proceed. So this step is for the filtration and the normalization is always the same as the single omics data. So for the microbiome data, it's same as the marker gene part. I just quickly processing it. And for the metabolomics data, we should have mentioned this in the statistical module. So we also need to do the filtering and normalization. We just keep the log transformation as we usually use in metabolomics data. The only thing need to mention is usually we will keep the auto scale as default to make the two omics data more comparable in a similar scale. Or sometimes there's a lot of zero in the microbiome data, then you can see the peaks are quite separate, then it's not comparable. So here is a PCA plot for the, we have a general idea, the data pattern in the different data set. It's similar as what we have seen just now. But next step, we would more likely to want to see if the paired data have like together to affect the group separation and help us to understand the biology. And before we go to navigate to the method page, we need first do the differential analysis. Here you can just specify the comparison group. For sure, here we only have this comparison. And if you want to do the control the covariant, and you can select the p-value cutoff, and in default we didn't specify the method here because we use a linear model like lemma for the metabolomics data, and then the Maslin-2 for the microbiome data, which are multivariant. We have more choice, and the result is usually better. And we can change the taxonomy level here. For example, we change to species, and the highlight are those species significant difference. Actually, these features are commonly seen in the papers related to the IBD studies. And for the metabolomics result is like here, this is peaks. Maybe if you can annotate this, which will be maybe better, but even use peak, we can look into some patterns and do the pathway enrichment analysis. So we can also build a box plot here like this. And then after this, we can go to the method summary. We have 3 big categories. First is dimension reduction. We only select 2 methods, Procrast and Diablo. The first one is unsupervised, and the Diablo is supervised. And the Procrast, which is a post-hoc integration. It's first do the analysis for separate omics, and then they combine the result and to explain. But Diablo will model the different omics together and to find the latent component. So they have the concept is different. So we may see some interesting result here. So firstly, we use Procrast analysis, and no, this is the Procrast, right? Yeah. So this we get the diagnostic statistics and figures. Here it's kind of clear to see we got the sum of square. This number actually means disalignment between the data. If it's high, like it's close to 1, which means the data actually not aligned well, which means microbiome and metabolomics data may be not aligned well according to the Procrast analysis. And you see the correlation of these 2 omics data. It's not too high, it's moderate. And then we got a significant result, like the significance, which means this result should not be random. It should have statistical power. And here we can see there's several, 7 samples, the data were not paired well. You can see here, in different groups, if the line is too long, means the 2 data not aligned well. So here is the summary. Then we go to the visualization part. It's a 3D scatter plot. Okay, then no connection is good now. So we can see there's different groups. They're not quite separate well, and we can see it's CD groups. Especially you can see the control groups. the data actually align better, but city groups is change a lot, which might meaning the disease group maybe have more like various, and it changes between the individual might be. And this is you can, I don't have time to show all the like a visualization part. You can explore to, we can add the background, no, add the floor or add the wall, or you can see the shadows here, because this is all the visualization technique. And you can also change the node style or the edge style, and maybe taxonomy, and download here. So you can explore more, use our example data, and then I will go to the Diablo. Diablo, because the modeling, it may take slightly longer than the Procrast. Okay, it works. So here we can see, as we mentioned, the Diablo is a supervised method, so we can control how many covariance we want to include in our model. So if the value is 0, it means it doesn't consider the covariance at all. It will maximizing the group difference totally based on the metadata. And if you increase this value to 1, then basically you won't consider the metadata. It's like unsupervised. It's just maximizing the covariance across the omics layer. And here we use the default, this 0.2 is a recommended value from the original group for develop this method. So we just keep it like this. And here we also see the diagnostic result. It says like the cluster error increase until the 5 component. In our result, we use the first 3 component, but the second and the third component still get a relative high error. But the first one, it has a very low error, which means the component is more reliable. And we can also see the sample space. It's for the top 3 latent. And we can see the correlation is quite high here. So based on the Diablo, we can see there's a very good alignment between microbiome data and metabolomics, which is kind of different from the previous result, which means because this method, the supervised method, it tend to align these 2 data together. So we can see there must be some signals that these 2 components have some alignment signals, but they might not the dominant one in the whole data set, which results the different analysis results from different methods. So it's based on your understanding. And then the third is loading. So basically in this example, we can see the first component is more reliable. And this is a biomarker, like selected top 10 significant features for either the microbiome data or the metabolomics data. This data you might want to look into more, because this might be the potential biomarker. Maybe you need to align this result with differential analysis or the functional analysis to see if they can really be considered as a potential biomarker. So this is the overall summary, and we go to the scatter plot. The plot, we have the same visualization technique as Procrast.
So can people download the loadings and the figures?
We can download figure. The loadings, I'm not quite sure.
Because that's something is that download here from.
Yeah, no, here not. Here download only. Then. Oh, you can just submit this to have a clear. So you can see the. It looks like the two groups through the Diablo, they kind of separate, and which means even it's not quite dominant, but the interaction between microbiome and metabolomics, to certain degree, they can help to distinguish between the groups. So this is the result for Diablo. So you have the similar visualization, and you can explore. The different thing is we have the result table here in the right side, and when you click, you can see the biplot to, for example, here, and you can see how the dominant features that drive the component. So you can see maybe we can change to a biplot view to see the did I select? Why it's not work? Maybe there is something wrong here. Okay. So yeah, not showing up.
That's fine. Can you just click some of the arrows from top left panel? And can you see that?
You mean this?
Yeah.
Yeah, for sure. So you can select the features to view how it drives the different components. Yeah. Do you have any question about this part? Because this. Okay. So if no, we can go to the next analysis. So next two, we both include the function part. This one was the customized network. Basically, because this sample, what we upload is the stool sample. Both metabolomics and microbiome is from the stool sample. So we think the metabolite mostly are from the microbe. So we can see if the peaks can enrichment in the metabolism of the specific microbe. For example, we can either use all the taxa we have uploaded or the significant taxa. So we just try what we have uploaded. So if, for example, if you upload like the girl mentioned, if you have serum metabolites, the explanation might be different because the serum, the metabolites, that's met from the host. So it's the integration might be different. It's not you directly project it to the microbe metabolism, because it's usually it should project to the human one. So that integration, like this part, should you need to pay attention how you explain the data. So here we get several pathways here. Clearly, like the several amino acids pathways, they are reported like a trypsin metabolism or the filamina metabolism. They all reported in the IBD study. So when you click here, for example, you see these metabolites, all these metabolites can be produced by the microbe. so which makes we are easier to understand the origin of the metabolites and the relationship, because we know like these metabolites why it change with microbe, because microbe may produce them. So the dots here like highlight the metabolites. If it's a circle with a green one, which means it's microbe specific metabolites that's not produced or consumed by human. But if it's red, it means it's like a common. It may share with human. Can be like a messenger. So this is.
You can double click on it.
Let me see. Oh, I can double click. So here, oh, I forgot this. All right. So here, when you click one dot, and you can see this plot, circle plot. So it highlight those how to say features, species that are most correlated to the peak, which means the origin of this peak potential metabolites may produced by this the most correlated microbes. So, but this is the statistical version. I will show like because this is peak, we cannot do the prediction because the prediction only for the metabolites. This is the circle plot actually the similar as this correlation, because here is a peak and the species correlation heat map, and you can see the stars here, which means significant correlation. You can change different methods. We have partial correlation, distance correlation. You can explore, and if you don't have too many samples, you may use the partial Spearman correlation, which is more stable for small sample, and remove the effect of other factors. And also you can change the color palette and change the threshold. Maybe you can depend on your needs. But this is a pity for the peaks, because we don't know if it's what metabolites it is. But if you have, I will show another example here. We have the metabolites. Then if you know that, then you have the microbe. You can overlay the two layers. First is the correlation, like the peaks. We get a statistical correlation. Then we have the GEM model to get the prediction. Like for example, here we see a diamond here. This one means the correlation between this metabolite and this microbe are both statistically significant and predicted by the GEM, which because in the GEM, like these species have at least more than 50% possibility to produce these metabolites. So this association might be more biological meaningful, and we can look into more details in MicrobiomeNet actually. So I think this is all for the integration part using MicrobiomeAnalyst. And next I will go to the MicrobiomeNet. This is kind of different with our other tools because it's a database tool, and as we show the design, we got a big database for the correlation, and then we look into those correlation, what's the detail, if they how the two microbes interaction. But if we are in real studies, like we study IBD, and we may have some specific species or metabolites we want to search, we want to know more. So we have some scenarios which we can use MicrobiomeNet. The difference is that in either MicrobiomeAnalyst or MetaboAnalyst, we have tables. But for the MicrobiomeNet, we only allow a single metabolite or a single microbe as input, because till this step we already have more understanding of our data. So we want to know more details, mechanical stuff. So we choose the most promising microbe to know their metabolism. So MicrobiomeNet can help us understand several questions I list for here. Maybe it's easier to understand, like whether a microbe can produce a metabolite and through which pathways, and the overall metabolic capacity of a microbe, and also whether two microbes interact with each other, if they are competitive or the complementary, and also if we find some correlations or associations between two species, if they have already reported in the same context in the literature. So we can find this information in MicrobiomeNet. So I just give an example because it's not from the previous analysis, it's just from the published studies, ABD studies, like we mentioned microbe, I will type it because I can't pronounce it. So which is a beneficial microbe of anti-inflammatory to produce like a short chain fatty acid. I will help it. I need to refresh. I need to refresh. So I need to type because I don't know how to pronounce. Sorry about that. So this is beneficial metabolites, like we mentioned a lot in the IBD, and also that this one. So we just mentioned this. So maybe in this we want to answer the question if how this microbe can produce or metabolize this short chain fatty acid. So we just search. Oh, well, I might type something wrong. I'm sorry. But I tested like a few minutes ago. It works.
The internet connection, no?
I have connection. Let me see, because I just checked. It's always work. Let me see. Oh, that's right. I don't know maybe I typed something wrong. So here we get this. It's like we can see there's two reactions involved in this microbe that can metabolize the specific metabolites. And in this pathway, maybe we can click here to see, maybe refresh. So this is how we highlight how the reactions that metabolize the specific metabolites. So this is this pathway in the species we are interested in. So this is the first question, and actually in this page you can explore a lot, but I don't think I have time to explain. So maybe you can test. We have the tutorial help you. You can find and explore yourself. And next, we have a gem view, which is the whole metabolism of this species. So you can click to view, but I will show this later in this map. So like this, yeah. I just this is a web issue, I guess, because when I here is quite slow. So here, this is the input species, and then we can search the other, like we find statistical correlation with this species. We can find it in the we can see highlighted here, and then it with a relative moderate metabolite distance, and but they have a quite different taxonomy distance. And this is all the data point highlighted here. They have been reported in the literature. So here you can find if the correlation you are interested in have been reported in the previous literature. We also have the table in other page, so maybe you can explore more. I just quickly go to the last thing is the complementary and the competitive between two. It's just like we can see here. So this is the input species, and this is the one we have explained. So we can see why they have already reported in the IBD study. They have association and have the anti-inflammatory function, and they are interact through the short-chain fatty acids. They complementary each other to both better growth and to when they are together. So this is a typical stuff because they have the relative high complementary index, which means they can produce the nutrients the other need. And then you can see the competition index. It's moderate. This is important actually because it's not like you are more complementary and less competition is most good, because if you have totally no competition, usually the two species cannot grow up in a similar environment. So if they can in the same context, it means they must have some shared nutrients to make them they can grow together. So most case is more meaningful is they have a median competition index, but they have higher complementary index. Then they may more complementary in a specific context. So when you click this, you can also see their complementary index. You can click to see the figure, and then you can also click the gem wheel to see the overall you can see their overall difference, because you can see the green one is for the bacterial, and the orange one is for this species, and the blue one is their common ones. So that's what we can find. Several things we can use MicrobiomeNet, and you can explore more. I cannot cover. Yeah.
Just can you bring up the slides?
Oh,
so
okay.
We are finishing up. I know we are last minute. I think hands-on, we do not have hands-on. So thank you for staying. And can I see you? No? Okay, so I guess we are right on time. Hopefully we don't have time to demo, to hands-on, but we do have all the detailed tutorials on the GitHub. Please do it if you are interested after you have access to online. And they also help us, and we have the training, and we actually have some pro version tools, especially you have large data needs. You want to save the project, and actually it's quite handy. And this is what it looks like. And actually, And the new one with AI, you can customize almost all the analysis and graphics. It's very cool. And if you have questions, post on Omics forum, and we'll be able to answer later. And then all of this is your TA and me. And so if you need support, and you can email us. But the best way is on the forum, just because a forum is open and everybody can benefit from that. Okay, thank you. Yep.